Cancer has nearly overtaken heart disease as the #1 killer in the United States. I'm going to be talking about just 7 top trends that I think are impacting cancer management and diagnosis, and some of the work that we do in our lab. So just talking about prevention and surveillance, if you look at lung cancer death rates, and so you have a curve here of lung cancer death rates. One thing you notice is that it has a defined peak here, about 1990, and falling. And pretty much the curve correlates with smoking rates. So as smoking decreased, you had a sharp decline in lung cancer death rates. But even so, you have about 15% of US adults who are active smokers, so an estimated 37 million Americans who are active smokers. If you include all former smokers who have a significant smoking history, up to about 100 million Americans. And this remains the #1 modifiable risk factor for cancer, and so lung cancer deaths remain over 150,000. If you add up the next 3 cancers combined, you have less than 120,000, which is prostate, breast, and colorectal. So it makes up more than a quarter of cancer diagnoses, as you see there. So lung cancer remains a big problem. More recently, we've run a very large clinical trial through the American College of Radiology Imaging Network, Akron, and this lung cancer screening trial was more than 50,000 Americans, and what we showed was basically that there's a 20% reduction in lung cancer deaths with low-dose CT. But this comes at a cost. So somewhere between $27 and $34 billion, the cost of screening and the downstream effects of screening over 15 years. And not only that, but it results in an estimated 30% of overdiagnosis. So these are real lung cancers that are going to be diagnosed, but they would have never killed the person. So this is the overdiagnosis estimated rate. And so the other thing to notice, as you can see in this graph over here, is that just very modest, so 10 times less investment, $3 billion investment in counseling and nicotine replacement therapies would have the same quality-adjusted life years saved. So think about that. We're gonna, as a nation, invest $30 billion or so into doing lung cancer screening with low-dose CT, which, as an interventional radiologist, and as a radiologist, it benefits my pocketbook. But that kind of cost, there's a lot of opportunity for innovation here, just in smoking cessation and behavioral modification. So can you automate that? The other thing to think about is, as the lung cancer has declined with smoking, we've also, though, had increase in other cancers due to obesity. So an estimated 100,000 Americans are diagnosed every year with new cancers related to obesity, and the obesity epidemic is such that close to 40% of Americans are obese. So this is a driving cause of fatty liver disease and liver cancer, which is one of the cancers that I treat a lot. So I think that there's a lot of innovation in that space around, if we can impact, obviously, obesity rates and smoking rates, then we can have large inputs at the very beginning in preventing cancer. So precision genomics. This is the idea that you could do a genomic analysis of a patient's individual cancer cells, and from that get actionable information in terms of which drugs you want to put them on, in terms of who needs treatment at all. Is this a cancer that is just going to be benign and not going to lead to anything? Or what dose of drugs, so pharmacogenomic kind of questions. And the real driver of this has been a project called the Cancer Genome Atlas. And this is something we refer to all the time in our lab. And basically what this is a large project cataloging the genetic mutations, kind of like a human genome project, but for cancer. And this repository of information, there's a central repository of information, a lot of collaborating institutions, ours being one of them. And what you have is kind of a Google Maps of cancer, not just looking at the genetic mutations, but looking at gene expression data, DNA methylation, et cetera. And the idea is that from this molecular profiling, you'll be able to tell which patients should be grouped together as different types of cancer. So one of the earliest things we found in our lab was that we have a biopsy protocol with liver cancer, and we biopsy the person's liver cancer, we take it out, and we do whole exome sequencing of their cancer. And many things that are called hepatocellular carcinoma phenotypically by a pathologist, when we run the genetics on it, it turns out that they're a sarcoma. It turns out that they're cholangiocarcinoma. There's some other thing based on the genetics when you compare it against the Cancer Genome Atlas, and that's what something like the Cancer Genome Atlas has allowed us to do very well. So obviously there's a lot of companies in this space, Foundation Medicine, Guardant Health, and now Grail, a ton of money flowing in there. Grail uses circulating tumor DNA to look at the genetics, but what they're all kind of in common doing is looking at that underlying genetic aberration and trying to get out of that functional intelligence for how to treat the patient. The issue that I have with this approach is that you can think of it as, cells have 20,000 genes, and maybe you have 100 to 200 mutations within those networks in the cancer cell. And you're somehow supposed to know all of the strengths of interaction and all of those genetic pathways to predict what drug you're going to be able to treat them with. I mean, this seems like, to me, it's a stretch. And so right now, there haven't been a lot of successes with that. And there are some exceptions we'll talk about. One of the other big things I'll talk about is patient-derived tumors. So one of our big precision medicine protocols in my lab is a thing where we do a percutaneous biopsy. We grow the patient's tumors in something called patient-derived xenografts. So these are mice that are capable of growing human tumors. We, at the same time, are isolating the patient's cancer cells, growing them in the lab, and then subjecting them to 2 different forms of screening. One of them is an old-fashioned kind of pharmacologic high-throughput screen with a chemical library where we actually take the approved compounds that are already out there. There's about 1,450 compounds that the FDA has approved over the lifespan of the FDA. So you can do just a high-throughput screening that way, or you can do our newer way, which I'm more excited about, and that's the CRISPR library approach. So it turns out that of those 1,450 compounds that the FDA has approved, those funnel through about 200 to 250 pathways. What you can do with that is you make your primers against that CRISPR library, you make a CRISPR library against this druggable pathways of FDA-approved drugs, and then by looking for synthetic lethality, where you knock out those individual genes, what you can arrive at is what is the one gene that cancer cell needed to survive. When you knocked it out, those cancer cells wouldn't live. And there now you already have, because you already know those compounds are already FDA-approved drugs, you have a method to get them into clinic very quickly. And then, of course, you validate them both in the cell lines as well as back in your patient-derived xenograft mice. So this is just some early data on a couple patients with HCC again. One of the first hits was something called ornithine decarboxylase. Now, I'd never heard of this gene. It's not something that I've ever run across in the cancer literature. But it turns out this drug, eflornithine, was developed back in the 1970s for cancer. It turned out to not be effective in clinical trials. And then it got repurposed for the treatment of trypanosomiasis, except for that Aventis, which is Sanofi Aventis now, was not making enough money from it in Africa, so they discontinued the product, and someone brought it back because they found out that it worked very good for postmenopausal female hirsutism, so hair growth on the face after menopause. Once they started making this drug again, it caused enough of an uproar in the literature that they actually started to work with the World Health Organization to make it again for trypanosomiasis, since they had already ramped up production for this other indication. So in any case, we took this drug, eflornithine, tested it out on that patient's cancer cells. Turns out it does kill the cancer cells. It does treat it in the mice. It does nothing to normal other HCC cell lines. And what's very interesting about it is is the fact that here's a drug that is being made out there in the clinic. There's no change in the ornithine decarboxylase gene. There's no mutations. There's not an increase in copy number. We have no idea why, in this particular patient, the constellation of mutations that they had made that cancer cell highly dependent on that gene, but that's the functional assay that you got. So instead of kind of looking for the needle in the haystack, you're starting with your picture of the needles that you have, and one by one pulling them out and seeing where they have their effect. The other thing to note here is that patient had no sensitivity to inhibition of RAF, suggesting that patient would not be a responder to sorafenib. Now, sorafenib is the one approved chemotherapeutic out there for liver cancer, and it's quite a toxic drug, and so you would not want to put a patient on it if you knew that they were not going to be a responder. On the other hand, this other patient, they were a responder to RAF, so that patient would probably be a sorafenib responder. So here we have a test that we can tell whether or not they're going to be a candidate for the one chemotherapy that's out there. In addition, that patient had a sensitivity to this hormone receptor, SRD5A3, and there's a drug out there, finasteride, for BPH. Again, it has effects on this cancer cell line, not on the other one. Do we know why? No, this is a functional screen where all you know is that you've pulled the gene out, and that causes the cancer cell to die. Artificial intelligence. So obviously there's a lot of work in the diagnostic space, companies like Analytic and Radiology, and there's other ones out there, that are basically doing image analysis using machine learning and artificial intelligence algorithms to pull out functional information for making diagnosis and making predictions about whether or not, for example, you should be biopsying that tumor or whether it's something that you can leave alone. There's a couple other companies in pathology states, Procia and Cernostics. These are doing the similar thing. Google runs an annual competition on being able to detect cancer using these kind of algorithms on a slide using pathology, and these companies are in that space. And there's actually one for looking at optical information coming out of optical colonographies, and that's this company AI4GI in gastroenterology. So in the therapeutic side, obviously Watson for oncology is probably the big elephant in the room. And that's what in my field a lot of discussion has happened around. Now, there's also a lot of criticism of this. Here was a recent article out of Stat saying, it pitched it as a supercomputer. It's nowhere close. And the claim in the article is that maybe it's more like a mechanical Turk, because in the essence, it's not synthesizing new information. What it's actually doing is regurgitating treatment algorithms that have been put in there by Memorial Sloan Cancer and Memorial Sloan Kettering and the other cancer centers that work with IBM on this product. But what I will say is that having used the product and having worked with others using the product is that it synthesizes, brings together a lot of the information that as a clinician you're making a recommendation on, but maybe you don't remember all the literature, maybe you haven't kept up with the latest trials on it. That's where this product, I think, comes in very handy when you're at a tumor board and you're making a recommendation, or if a patient's asking you, why did you choose that treatment over this treatment? This is kind of hand-holding for you a lot. So I think that it has its own functional role. Maybe it's not to the point of synthesizing new treatment paradigms, but it definitely has a role in cancer therapy now. And we're going to see a lot more of those kind of products. Let's talk about targeted therapeutics. Gleevec, that's the original targeted therapeutic, wonder drug for CML. It was a very specific tyrosine kinase inhibitor. You notice that the IB, all these IBs are tyrosine kinase inhibitors. But now there's a whole host of them for lung cancer, one of the cancers I treat a lot of, erlotinib, gemfitinib, et cetera. And a lot of this is really built around, believe it or not, one single mutation in the EGF receptor, T790M, which makes patients resistant to these drugs. But with third-generation mutant-selective drug, osimertinib, these patients are actually highly responsive. So what will usually happen is the patients will go on these first- and second-generation drugs. Once they clonally select for this guy, they go on the third drug. So these are targeted therapeutics. Obviously, immuno-oncology is big in our space. Dendritic cell therapies and adoptive T-cell therapies. We'll talk about, the next speaker will talk about CAR T-cells, and we do some of that work in our lab. And also checkpoint inhibitors, which is probably the lion's share of the market in immunotherapy right now for many different solid malignancies, including recent approval in hepatocellular carcinoma. Also, of course, targeted therapeutics and nanotech. There's not a lot of clinical trials out there with this, but there's definitely a lot of translational research in this space. This is one of my favorites in the past year, which is something called Cornell dots, these C dots, which are silicon nanoparticles that get taken up by the lysosome of cancer cells and lead to reactive oxygen species, which cancers are very sensitive to. And they undergo, this is a great name, ferroptotic cell death, so kind of like apoptosis, but using iron. Image-guided therapies. I'm an interventional oncologist, so a lot of what I do is, people talk about the augmented reality or virtual reality world. I never actually see the cancer, except for maybe a biopsy I take out. I never am actually looking at it. What I'm looking at is, of course, a patient in a big camera, like a CT scanner. I'm putting my probe into the tumor, in this case, a tumor that was sitting right on the aorta. Put my little probe in there, froze it a little bit, pulled it away from the aorta, and then treated that cancer. The patient was awake the whole time, went home with the Band-Aid. So these kind of image-guided therapies, you're going to see more and more of these minimally invasive image-guided therapies out there. We've been recently experimenting in my lab for complex cases and complex anatomies, looking in the three-dimensional space and augmented reality with HoloLens. And so occasionally for my CT-guided procedures now, my colleague who's making these data sets for me will put this into the HoloLens 3D space, and I can actually look at the patient and see the tumor right through the patient's body, the CT projection inside of them. And so it allows me to get a much better three-dimensional idea of the treatment that I'm getting ready to do. This is a great thing out of another lab at Penn, the single lab. And what you're seeing here, here's a tumor in the lung. He went in there to take this tumor out. And what he's going to do is he's injected isocyanated green, which is a dye that fluoresces under certain types of light. And here's a cancer that you did not see with your naked eye, but then became present and easily visible with that sort of optical imaging. So you're seeing a lot more optical imaging, molecular imaging agents hitting the standard surgery market, and you're going to see more and more use of that. So finally, I wanted to talk about patient-centered oncology. There's been, my patients show up to me all the time with clinical trials. And it's just, they are very, very motivated. The patient community, obviously, is very, very motivated for survival. So they all network through things like I Had Cancer. There's cancer-specific social networks. There's advocacy groups that bring people together and that provide funding to labs like mine, as well as obviously harnessing the patient population towards it. There's a push towards home diagnostics. Most of my patients would much prefer that we were doing their follow-up liquid biopsies and stuff at home. And one of the big breakthroughs I've seen in the past year has been this is a very simple little device that separates urine exosome, so extracellular vesicles that you're peeing out and that have cancer markers on them. The question has been, how do you isolate them? And I have isolated these with centrifugation in the lab, but it's quite a laborious process. And so these guys, I think it was out of Korea, made a small device, a little spinning disc that separates the urine vesicles, extracellular vesicles, and then you can do assays on them. So this tech has not been brought into the United States yet, but I assume that this will be part of any kind of Qualcomm tricorder that we're working on. So I think that there's a lot of innovation in that space. And then finally, the patient-centered outcomes. What do patients really want? And in many of my cases, I'm dealing with palliative procedures. Patients who are at the end stage of life and dealing with terminal pain. And so the outcomes that the patient's asking for may be different than what you as an oncologist think that you should be treating. And so I think that more and more we're seeing these patient-centered outcomes. So that's kind of some top trends. I didn't really talk about cancer stem cells, which an earlier speaker mentioned, and there's a lot of other cool stuff going on in oncology. But these are just some of the trends that I wanted to bring up that we're working on in our lab and looking at in that space. And thank you very much.
Thank you.